Hey there, and welcome AMP2 students to part 3 of renal physiology. In this video, we will look at how the kidneys are able to concentrate urine through the countercurrent mechanisms, and we'll talk about the hormones that are involved in that process. Knowing these processes is crucial to understanding how certain medications work to manage both blood volume and blood pressure, so pay close attention to the information given in this video. To truly understand these mechanisms, you must have a good grasp on the concepts of concentration, osmolarity, and osmosis. So if you feel like you need a refresher from Chapter 3 on these topics, pause the video. Okay, so let's recall that the normal concentration of blood and tissue fluids is around 300 milliosmolar, and that the passive movement of water and or solutes will always be in the direction to equilibrate or equalize these concentrations. And so depending on the permeability of the membrane to these substances, they will move along their chemical and or electrical gradients. Also remember that permeability is related to a substance's lipid solubility. If a substance is not lipid soluble, it will not be able to move through the cell membranes very easily. In these cases, the cells may have carrier or channel proteins in place to allow these substances to diffuse across. And if a cell needs to, it may also drive substances against their concentration gradients across membranes, and this would require a carrier pump protein and ATP. So there's nothing new going on here. We saw all of these mechanisms occurring with reabsorption, and hopefully by now you're understanding membrane transport. Let's see how all of this plays into how the kidneys concentrate urine. I want to review some important points about the microscopic anatomy of the kidney that will aid us in understanding this physiology. Remember that there are two kinds of nephrons, the cortical nephrons and the juxtamedullary nephrons. The cortical nephrons have short loops that do not dip very far into the medullary region. On the other hand, the juxtamedullary nephrons have extremely long nephron loops that extend into the deepest part of the medulla. Now, the peritubular capillaries of the juxtamedullary nephrons are specialized into the vasa recta, and they are intimately connected with the nephron loops. The juxtamedullary nephrons make up only about 15% of the total number of nephrons. However, they are critical to the process of concentrating urine and conserving water. In this picture, we can see that the collecting ducts also extend deep into the medulla. And they also are crucial to this process, so we will be spending the majority of our time looking at these structures. There are a number of key points to make about the design of the kidney that allow it to be such an efficient water-conserving organ. So I will run through these and then we will discuss how these conditions are set up. The first point is that the environment surrounding the nephrons, which we will call the interstitium, changes as we get deeper into the kidney. As we travel from the cortex to the deep recesses of the medulla, the concentration of the interstitial fluid increases to a maximum of 12 to 1400 milliosmolar. If you would eat a kidney from the outside in like you would an apple, it would get saltier as you got to the core. We will discuss why this gradient is so important for the kidney to be able to conserve water. The second point is that the long loops of the juxtamedullary nephrons have fluid in tubules that run in opposite directions to each other. The descending and ascending limbs of these loops are in close proximity with each other, as well as the loops of the vasa recta. Thirdly, the medullary collecting ducts are located alongside these nephron loops and are subjected to the same gradient so that their environment gets saltier the deeper they are in the medulla. And the last very important point that I want to make is that the permeability of the collecting duct, both in the cortical portion and the medullary portion, as well as the distal convoluted tubule, is under hormonal control. With all that in mind, let's take a look at how the kidneys have set up the medullary salt gradient and how it uses it to produce either a dilute or a concentrated urine. Looking closely at the nephron loop, we have the descending limb coming from the proximal convoluted tubule, and the filtrate will be traveling down toward the turn of the loop. Now, the descending limb is highly permeable to water because of the presence of aquaporins. and water will move out of the tubule, equalizing the osmolarities inside and outside the tubes. So the osmolarity of the filtrate is 300 milliosmol as it enters the descending limb at the beginning part of it, and it gets more concentrated as water diffuses out, such that the concentration within the descending limb will match the surrounding medulla interstitium. And the descending limb is impermeable to solutes, so they stay behind in the tubule, concentrating the filtrate. As the fluid rounds the hairpin turn of the loop, it's at its most concentrated of about 1200 milliosmoles. And on the trip back up, things change. Things are a little bit different. In the ascending limb, the cells are actively pumping the solutes out, but the tubule is impermeable to water. So water does not follow the solutes, and the fluid within the tubule becomes less concentrated as it ascends toward the distal convoluted tubule. As a matter of fact, by the time the fluid enters the distal convoluted tubule, it gets so dilute that it is hypoosmotic at 100 milliosmoles. This is a critical point. The solutes that are being pumped out of the ascending limb will serve to produce the gradient of salt called a salinity gradient that we see in the medulla. You might be asking yourself, well, what about the water that is leaving the descending limb? Isn't that going to wash out the gradient? Well, that's a good question, and we will take a look in a bit as to why that does not happen. So what we have here is something called a countercurrent, which just refers to the movement of the fluid flowing in opposite directions in the two limbs. And we have the pumping out of the ions adding to or multiplying the gradient. We call this process the countercurrent multiplier. Just how this gradient gets set up to begin with is a fascinating topic, but one that we will not get into at this time. So the pumping out of the solutes from the ascending limb helps to maintain the gradient, and we want to look at how the gradient is not lost through the washout. So in the juxtamedullary nephrons, the peritubular capillaries that branch off of the efferent arteriole, we see them as the vasa recta. This is actually another countercurrent system. Blood flowing from the efferent arteriole is isoosmotic to the surrounding cortical interstitium at 300 milliosmoles. The capillaries of the vasa recta are permeable to both water and solutes, so the vessels will pick up an excess of either one of these. Also remember that these vessels contain the blood coming from the glomerulus, so the hydrostatic pressure is low and the colloid osmotic pressure is high. And these conditions favor reabsorption into the bloodstream. If these were straight vessels, the water and solutes that were reabsorbed would be just washed away and transported away from the area. However, because they are also configured as loops, the majority of the solutes will be returned to the medullary interstitium and also a small portion of the water. The solutes will cycle in and out of the vasa recta continually, being restored to the interstitium instead of being transported away, as we see in this picture. There's a similar recycling of urea that occurs between the collecting duct and the nephron loop that we can see in this picture. And this contributes to the salinity gradient as well. As a matter of fact, urea is responsible for about 50% of the gradient. So basically, the two limbs of the vasa recta exchange the water and solutes, maintaining the gradient, and this is called the countercurrent exchanger. Now let's put the two countercurrent mechanisms side by side and see how they all fit together. Be aware that our examples are far from realistic. There would be many nephrons and vasa recta associated with each other, contributing to and affecting a single area of the medulla. So don't be fooled by our oversimplification. Now spend some time with this picture and realize that the vasa recta are in more intimate contact with the nephron loop. And if we turned it around like this, we might be able to get a better sense of the directions that things are moving. We'll look at this again in class, so be ready to have a conversation about it. Our final discussion will be about what happens to the fluid as it enters the distal convoluted tubule and the collecting duct. The kidneys will be making their final adjustments to the fluid, transforming it into the urine that will be excreted. The two hormones that control what goes on at these tubules and that we're going to be considering are aldosterone and antidiuretic hormone. First, we will look at what happens to the fluid when neither hormones are present. So the fluid entering the distal convoluted tubule is hypoosmotic at 100 milliosmolar, and it will pass into the collecting duct relatively unchanged, leaving the body as a dilute urine. Now, under the influence of antidiuretic hormone, things are a bit different. Antidiuretic hormone stimulates the tubule cells of the collecting duct to produce aquaporins. Since water will be able to leave the tubules, it will do so and equilibrate the osmolarity of the urine to the surrounding medulla. The more aquaporins that are present and the further down that they are placed into the collecting duct, the more concentrated the urine can become. Now, the water reabsorption that is occurring at this point, which is dependent on the presence of ADH, is known as facultative water reabsorption. The conditions under which ADH will be released are when we're dehydrated and when our blood volume and pressure are too low. The body conserves water to increase the blood volume and the blood pressure. Now, aldosterone's effect on the kidneys is also to conserve water, but in a different way, and it is nowhere near as effective as ADH in increasing blood volume. Aldosterone targets the distal convoluted tubule and the collecting duct to synthesize more sodium and potassium channels and carrier protein pumps. This causes more sodium to be retained by the body and more potassium to leave in the urine. And there is a one-for-one relationship here. For every sodium that is conserved, there's a potassium lost in the urine. Now, water will follow the sodium if aquaporins are present. You might be asking yourself, why would water preferentially follow sodium and not potassium, since there's a one-for-one exchange? The reason for that is because chloride follows sodium, and that sets up an osmotic gradient for water. All right, that should give you the information that you need on how the kidneys are able to concentrate urine and conserve water. Please read about the different kinds of diuretics on page 857 of your textbook and exactly where and how they have their effects so that you'll be ready to have a conversation about this in class. All right, see you all then.